Thank you very much. Our ladies, Dr. Borini, thank you for these amazing meetings. It's very exciting. And what I would like to talk to you today about is a little bit about one of the key questions for fertility. And that is really what happens to IVF children when they will grow up, when they will be in their 50s and 60s. Will they be healthy as we are? In this talk, I will introduce briefly the so-called Barker hypothesis and then show some animal data regarding effect of culturing an embryo in vitro on long-term health, and then specifically analyze gene expression in blastocysts to see if we can correlate events that occur in blastocysts to long-term health. So the question is, we know 5 million children have been conceived, but are there really problems long-term? One of the key studies that we all should be aware is the study by Dr. Schultz in Pennsylvania that showed that simply culturing embryos from the two-cell stage to the blastocyst stage was associated with difference in behavior. Mice culture for 2 days in vitro had decreased memorization of spatial information and decreased anxiety. Therefore, this study published in 2004 and confirmed by Dr. Fernandez Gonzalez in Spain showed that clearly there can be problems that we don't know yet. So the true question is what happens here down the road when the kids will be 50 or 60s? The advantage of the mouse model is that mouse lives approximately 2 to 3 years, and therefore at the age of 6 months to 1 year is like having children at the age of 50, and therefore we can discover events way earlier. Of course, we'll need to be here in 2042 or 2052, 30 to 40 years from now, to know if really something will occur, because only epidemiological studies will really tell us if there is any problems. The intellectual background for these events is the so-called Barker hypothesis. Dr. Barker in the 80s found that area of Sussex, Wales, and Lancaster where high level of heart attack and stroke were present were also the same area where children were born of very low birth weight. So it created an association between low birth weight and event happening later in life, therefore suggesting that what happens in utero has long-term consequences. In utero event can have really important effect later in life. This is a well-known phenomenon even in animals. Just to give you some example, if you have crocodile eggs, at 30 degrees they will be all females, at 33 degrees they will become males. So the organism senses the environment during development and reprograms itself to different strategies. For example, in this particular insect, the African armyworm, if there are low nutrition, it will completely shift his metabolism from carbohydrate utilization to triglyceride utilization. So it is an example of how, during development, the organism can sense what happens and modify the growth. So the true question is, do we have long-term health outcome? What do we know of the current evidence in ART children? We know that 5 million children have been conceived, and overall, they're very healthy. The current evidence in singleton, beware, show that overall, there is a slight increase evidence of preterm birth, or at term, low birth weight, very low birth weight, or intrauterine growth restriction. So even singleton at term, suggesting that something happens in utero following ART. And in a big meta-analysis by Dr. McDonald comparing 30,000 ART children to 80,000 control, they found that overall ART children has slightly lower weight, 97 grams less, and slightly shorter gestation. However, this is important because, again, if birth weight is a marker of stress, this could indicate that something has occurred during development. Yesterday, we have heard from Dr. Huntress that imprinted genes disorder are also frequent. And indeed, imprinting disorder could be a marker of abnormal methylation and problem to come. What about the published evidence on long-term health of children? There are very few studies. In my mind, the best studies derive from the Netherlands, where Dr. Céline Manon compared health of 233 IVF children with 233 control children that, however, were conceived by subfertile couple, meaning after 1 year of trying to conceive. And therefore, is a very good control, because these are infertile patient who conceive naturally. And so it's really probably the best study out there. Importantly, there were no difference in behavior. However, there were some difference in peripheral body fat deposition. And at age 12.6, however, slightly higher blood pressure, slightly higher glucose level, very, very minimal. That doesn't have any clinical significance at age 12. However, the question is, could this be that later on in life there will be problems? So given this limited human evidence, what we've tried to accomplish in the laboratory is try to see if can we find any evidence of metabolic problems later in life. So we follow mice and we culture them in vitro, we perform embryo transfer, we see what happens. And we use two particular culture conditions. We use the so-called Witten's medium, that is a suboptimal medium, was developed in 1970 by Dr. Witten's, and the optimized medium is the so-called Cason medium. It was developed in 1993. And Cason medium is the basis of multiple human IVF media used today. But if you notice, the difference between the two media are very minimal. A little bit less glucose, 0.2 millimolar versus 5.5 molar, 1 millimolar of glutamine, and presence of amino acids. Therefore, very little difference. So we perform in our experiment in vivo conception and follow animal as a control. The advantage of animal models that we can have a control, and number two, these are fertile animals, so the concept of infertility is not there. And then we created a dose response, more stressful condition or less stressful condition. So the first data are postnatal growth curve using Witten's medium, that again is the most stressful medium. Here you can see the growth up to 40 weeks of life. And the green is the control, the red is the IVF mice. And they have a lower growth for the rest of their life, right here. So they're lower in growth. This is true for both male and female. And so importantly, these mice in worse condition have a phenotype that grow less for the rest of their life. And second, when we perform a glucose tolerant test on these mice, they are glucose intolerant. You perform a glucose tolerant test the same as you perform in pregnant ladies. You give glucose and then you measure glucose at different interval. Now, what we see both in male and female, that the smaller IVF mice, importantly, are glucose intolerant. As you know, obesity is associated with glucose intolerance. The fact that even the leaner IVF mice have this phenotype suggests that there is really an event occurring during development. The second set of experiment instead was performed using optimized media, the one that we use in IVF, and also we culture embryo 5% oxygen. The good news is that we saw very little effect. Overall, the growth curve was no different between IVF and control. However, we saw some different female mice. Female mice start to become heavier at approximately 15 weeks of gestation. We follow up with glucose tolerant test at 13 weeks, 19 weeks, 21 weeks of life. No difference, so good news. However, there was slightly higher level of insulin in female mice, and the fat content in female was higher. That was consistent with the human study that I quoted before. So basically, what we saw with this phenotypic experiment, we saw a dose-response effect. Optimal condition have a minimal phenotype close to normal, however still present. Suboptimal condition, a very clear phenotype. So the question is, what is the mechanism? And obviously the preimplantation embryo undergoes a series of development that are very unique and important. For example, there is zygote genome activation in the two-cell stage, blastocyst formation, cavitation, and importantly, there are changes in methylation throughout the stages of development. And again, as we have seen yesterday in Dr. Huntley's talk, that is very important. Imprinting disorder are just an example of potential abnormality in methylation present in these mice. So we decided to ask the question, can we understand if the gene expression pattern in blastocysts can give us an idea of why we found that particular adult phenotype? Or more specifically, we have these, can we correlate to blastocyst gene expression? Can we learn something from microarray analysis? So before going into the result, I want to briefly explain some of the results that you will see. There will be multiple clustering analysis studies in which here, for example, is an example of good clustering in which every repetition indicated by the color are together. So the intra-group variation is very small, the group clustered together. Here is an example of poor clustering with minimal changes in gene expression. When you see these, this means that overall there were not many differences in gene expression. When you see that, it means that the gene expression really marked between the groups. The second graph that you will see moving forward will be about gene expression. You will see a graph like this in which there is a mean, and then there are changes in gene expression going up or down, so the number of genes that are differentially regulated. And here you will see bands of different color. Here, the first one means all the statistically different genes, and next are gene changes more than twofold or more than 4 or 5-fold, depending from the graph, and then there will be different groups. So the first series of experiments was simply to ask, okay, let's simply culture zygote fertilized in vivo to the blastocyst stage in different media or in different media with different oxygen concentrations. Again, we use Witten's and Cason medium. And the first result, this is done using Witten's and Cason, the first result is here in this clustering analysis. There was not a big difference in gene expression, okay? There were the in vivo embryos separated in a separate cohort, but the different culture media, there were not so many different to differentiate each group among themselves. And overall, there were only 114 genes different between Witten's medium and in vivo embryo, and 29 genes between Cason medium and in vivo embryos. However, when we do other experiments using 20% oxygen, as you can remember, we breathe currently 20%, but in the genital tract, the oxygen concentration is 5%, so it's the physiologic oxygen concentration. Now, during this condition, we have up to 10-fold of gene expression changes. We had 114 genes different in the blastocyst. Here we have 1,200. And there we had approximately 29, and here we have approximately 300 different genes. So it's remarkably different. The effect of oxygen, using atmospheric oxygen to culture embryo, has remarkable effects. And I think this is important for practical purposes because many laboratories still grow embryo in 20%. And so this would be an indication to suggest that we should culture embryo in lower oxygen. So what we have learned from this experiment, well, we have learned that there is an association between phenotype and gene expression. Look at this. In these mice that have a glucose intolerance phenotype, we had a remarkable different gene expression. Approximately 1,000 genes were different right here, and then we have a phenotype. In the other group where we had a mild phenotype was this one over here. Only approximately 30 genes were statistically different, okay? So the first information is truly that the stress in the preimplantation embryo is important and has long-term consequences. And here you can see also actually the number of genes that are different. Overall, only 0.3% of genes were different at the blastocyst stage in this group, while here 13% of genes were different. And so consequences are real. So the conclusion number one is that culture of a preimplantation embryo in more stressful condition lead to progressively worsening gene expression. The gene expression go from 0.3 to 13%, and I removed some slide regarding what kind of gene are changed, but overall ribosomal and molecular transport genes are differentially expressed. The second question is now let's perform in vitro fertilization because this is clinically relevant. However, we decided to use the stressful condition. The reason to do so is because we wanted to assess if you could find a mechanism and stressing the system more, we could learn more about what is wrong with the embryos. And so the very first important result from the clustering analysis is that the three group, in vivo embryo, in vitro culture embryo, or IVF embryo, cluster very separately, suggesting that even the first 24-hour in culture have a profound effect in development. I would have argued before doing the experiment that we would have seen something like the culture media, that the group between in vitro culture and IVF were not clustering well together, but instead they really separate well, therefore suggesting that there is a clear effect also of the first 24 hours in culture. Then if we look at the number of genes, there are a lot of genes that are statistically different. Thousands of genes are different between IVF and in vivo, and also, interestingly, between IVF and in vitro culture embryo. So suggesting that every specific event that we do, how we manipulate the embryos, has effect and consequences. Regarding the classes of genes that are statistically different. There were multiple that were some upregulated, multiple downregulated. And importantly, there were downregulation of fat cell differentiation and some of heart forebrain bond mineralization development. As you remember, there was a normal fat deposition in IVF children, so this could be potentially a mechanism. So conclusion number 2, surprisingly, IVF embryo were different from in vitro culture embryo. Although there were multiple genes that were different, the gene expression changes were overall small. And finally, there were multiple classes that were different, proliferation, apoptosis, or morphogenetic pathways. The third question is ICSI. Currently, in the United States, approximately 70% of cycles use ICSI. Obviously, the male factor is probably 25 to 30 or 40%, but definitely not 70%. But it became a very common procedure. The question is, what happens? In this series of experiments, we culture embryo either in caseum or in Wittens, and here I'll show you just some data about the total number of cells that compose the embryo. T are the trough ectoderm cells that will form the placenta. ICM is the inner cell mass. And what you can see is that culturing an embryo, ICSI embryo, have less inner cell mass cells and less trough ectodermal cells, suggesting that the ICSI procedure in itself affects the morphology and the quality of the embryos. When we perform, obviously, IVF in humans, we don't have a control. We cannot check what happens when a blastocyst in vivo. Okay, the value of mouse or rodent or animal data is truly allowing a good comparison. And importantly, there was a little bit of a surprising result. The fact was that overall, either caseum or Wittens, the two media have a very high effect on gene expression, but the two media themselves were very similar among themselves. Again, before doing the experiment, I could have argued that, like in the oxygen experiment, we would have seen an amplification of the effects. Instead, here, we see that independently of the media used, the method of fertilization, ICSI, had a more profound effect on determining the gene expression. Also, the second information that there are a lot of genes were highly misexpressed. Usually the gene difference were 1, 2-fold. Here we have hundreds of genes different, like 200-fold. 200 genes were changed more than 5-fold. So abundant difference in the amount of gene expression. So conclusion number 3, basically blastocysts resulting from ICSI have a reduction of inner cell mass and trough ectodermal cells. Approximately 1,000 genes are different, but many of these are changed more than 5-fold. And unexpectedly, only 41 genes were different between the two culture conditions. The fourth experiment was, okay, the blastocyst is indeed form of two tissue, trophoectoderm and inner cell mass. What happens if you separate the two tissue? And then we can exactly compare the gene expression of the inner cell mass to the trophoectoderm in vivo and in vitro embryo. So the first set of experiment, we asked the question, okay, let's compare the difference between the inner cell mass of in vivo embryo and the trophoectoderm of in vivo embryo, or the same for IVF embryos. And the interesting part is that overall, the gene expression difference between the inner cell mass and trophoectoderm of IVF embryos were minimal. Okay? Instead of being amplified, it seems like the two tissue didn't differentiate as quickly as happens in vivo. And second, there were no different imprinted genes. We know the difference should occur. It's known that, for example, H19 and IGF2 are supposed to be different between tissue. And here, the expression, again, these are not methylation data, but simply expression data, they were not different. Then we compare the gene expression between ICM in vivo and ICM in IVF, and we found that overall there were 300 genes different between the IVF inner cell mass and the ICM in vivo. And importantly, some genes were instructive of what could be a mechanism. For example, BMP4 was downregulated in the inner cell mass of IVF embryo. Now, BMP4 is important for trophoectodermal cells differentiation. If you have low BMP4, you will have low trophoectodermal cells. And that indeed is what we found counting cells. And secondly, the gene expression of the trophoectodermal cells of IVF showed decrease in transporter genes and decrease of genes involved in implantation. In addition, there were some unique imprinting genes that were misregulated, like CD81 or H19, suggesting again that methylation events have occurred and could explain the mechanism. So the conclusion number 4, doing a specific analysis of the transcriptome of in vitro produced inner cell mass and trophoectoderm, is that there are few differences between the two tissues following in vitro culture. In addition, the trophoectoderm of IVF embryo showed decreased gene expression of genes involved in placenta formation, like EOMs and SOX3, and gene involved in, in implantation, like epidermal growth factor. So that could explain why we see some of the phenotype described. So in summary, what have we seen? We have seen that overall, the preimplantation embryo are exquisitely sensitive to each manipulation that we do, to the culture media we use, the oxygen concentration, the type of fertilization method that we do. And therefore, each manipulation generates unique changes in gene expression and potentially, I argue, long-term consequences. Therefore, there is the increased manipulation might result in increased abnormality long-term. So what is the long-term message or the take-home message? The message is whenever possible, whenever feasible, we should try to manipulate embryos the least. This is consistent also to the work of Dr. Lee's, the so-called quiet embryo hypothesis, meaning that the healthier embryo are the ones that are stressed the least. And therefore, whenever feasible, because I'm very aware of the multiple risks and PGS and etc., but whenever feasible, we should always strive to minimize manipulation to the embryos. And now, the most important slide is thanking all the people that have been involved in the study. Luisa Delle Piane, who is in this audience, was very important in some study that actually didn't have time to show. Marlea Di Santo is doing some work on stem cell currently, and Giri and Xiaowei performed the microarray study on embryos. So thank you very much. And of course, Andrea for the great support.